Hello, my name is Dr. Nicola Much, and I'm here to talk to you today about the interplay between fibrinolysis and platelets. Before we start, I just want to thank the organisers for the kind invitation to speak at this thrombin conference. And I also want to make a special thank you to the people who've contributed the work that I will be presenting today. That's Dr. Claire White and Dr. Claire Morrow, who are current postdocs in my laboratory, and Dr. Joanne Mitchell, who was a PhD student in my laboratory and is now a postdoc at the University of Birmingham. So we all know that hemostasis is the balance between fibrin deposition and degradation, but we often hear a large part of the emphasis placed on the fibrin deposition part, with little on the removal of fibrin from the circulation. Platelets act as a focus of fibrin formation, therefore it is perhaps not surprising that they could potentially influence fibrin degradation. If we think about the various components of a platelet and how they contribute to fibrin formation, we know that they expose phosphatidylserine on their surface, which then allows them to bind various coagulation factors and promote thrombin generation. They also house a number of coagulation factors within their alpha granules, including factor V, factor VIII, fibrinogen, and von Willebrand factor. They also have various receptors on their cell surface, and one of these, alpha-2b beta-3, is sometimes termed the fibrinogen receptor, because fibrinogen can bind to this integrin and potentially act as a molecular bridge between platelets within the aggregates. The ability of platelets to stimulate thrombin generation and subsequent fibrin formation can be visualized in this microfluidic model, where we have labeled platelets with DIOC6 and we have added fluorescently labeled fibrinogen. And you can see that fibrinogen just forms immediately adjacent to the activated platelets. And actually you see some directional deposition of the fibrin in the direction of flow. Platelets have largely been considered antifibrinolytic in their function, and that's because they are known to house the major circulating pool of the SERPIN inhibitor, plasminogen activator inhibitor 1. This diagram here just shows where various cell types that can synthesize PI1. And in terms of the vasculature, the endothelial cells are a major contributor to PI1 synthesis, but also within the circulating component of the vasculature, platelets, as I say, are the major contributor of PI1. The PI1 gets packaged into the alpha granules during platelet production, but platelets also house PI1 mRNA and can go on to synthesize further pools of platelets during their lifespan. It's been well known that platelets secrete PI1 on activation for many years. It was first discovered in the 80s, actually by my mentor, Professor Nuala Booth. However, some of our recent work has shown that as well as being secreted into the supernatant, we also find PI1 is abundant on the platelet surface. So when we activate platelets, and this is when we've laid platelets down, washed platelets on a collagen thrombin surface, we can see generation of both spread platelets and procoagulant platelets. The procoagulant platelets are detected with annexin 5. And the PI1 is stained in yellow here. This is vitronectin, PI1's cofactor, and you can see that we have binding or localization of the PI1 on the activated platelet membrane. This is shown in a little bit more detail here, where we've got a zoomed in image of the activated platelet and the deposition of PI1 in what's called the cap region or balloon of the balloon-shaped platelet. The staining on PS negative platelets is a little bit more diffuse over the entire surface. And what we see, this is flow cytometry results shown in the graph here. The top graph is percentage Pi1 positive platelets. And as we stimulate those platelets with stronger agonists, we see more exposure of Pi1, but dramatically the mean fluorescence intensity is increased when we stimulate with dual agonists, in this case convulsant and thrombin, which will hit both GP6 and the PAR receptors on the platelet surface. So there is much greater retention of Pi1 when we expose the platelets to strong agonists. We can monitor the impact of that on fibrinolysis by confocal microscopy of platelet-rich clots. And you can see here that the top panel, which represents lysis induced by TPA, we can see movement of a lysis front over time. And when we include a neutralizing antibody to Pi1, we can dramatically enhance the time to lysis, indicating that Pi1 is active in resisting TPA-mediated lysis in this context. But as well as Pi1, platelets contain additional inhibitory molecules, and our laboratory has shown that there is a exposure of a transglutaminase called factor 13 on the surface of activated platelets following strong stimulation. Factor 13A was known to be abundant in platelets and was first described by Laszlo Mosbeck, but as this transglutaminase lacks a signal sequence, it was not known how it actually was released from the cells, because it's contained not within alpha granules as Pi1, which I just described, but is instead within the cytoplasm of platelets. So it was very much thought to play intracellular functions. However, we were able to show that while when we activated platelets, factor 13A became externalized to the outside of the platelet membrane, but interestingly was not secreted into the supernatant. And this is also true that we see exposure of factor 13A on spread platelets. We see similar results with confocal microscopy when we unstimulated plates have low levels of factor 13A, and this can be increased post-stimulation. And likewise, we see disappearance of factor 13A from the cytoplasm when we stimulate and exposure onto the membrane surface. I won't go into detail as to how this platelet factor 13A functions, but suffice to say that we have previously shown that plasma factor 13A is able to exert antifibrinolytic function primarily through cross-linking of alpha-2 antiplasmin into the fibrin matrix. And alpha-2 antiplasmin is the principal inhibitor of plasmin in the fibrinolytic system. And that's very similar in terms of the function of platelet factor 13A. And you can see here, if we have plasma in the red line composed of factor 13A depleted plasma, they're quite unstable, with fluorescence release being equivalent to the degree of lysis. But when we include platelets in this context, we can stabilize these thrombi against lysis. But if we neutralize alpha-2 antiplasmin, this stability of these thrombi is lost. And the same is also true when we inhibit transglutaminase activity directly, indicating that we require both functional factor 13A and alpha-2 antiplasmin to observe this antifibrinolytic effect. There is other fibrinolytic inhibitors that are present in platelets, and I'm not going to go into these in depth today because this is not work that stems from my laboratory, but you'll see here a very nice publication from Martin Jandreau-Perousse's laboratory that showed that protease nexin 1 can exert an antifibrinolytic effect by inhibiting TPA, thereby downregulating fibrinolysis. And it was also being shown by the group of Laurence Mosnier that platelets contain procarboxypeptidase inhibitor, thrombin activatable fibrinolysis inhibitor, and this was also shown by the group of Michael Boffa. Now, when we think about fibrinolysis, we think about fibrin being the classic surface for plasmin generation. And this is perhaps not surprisingly so, in that we know that TPA and plasminogen bind very avidly to the fibrin surface. And in fact, TPA mediated activation of plasminogen to plasmin is stimulated over 1,000-fold in the presence of fibrin. So fibrin therefore potentiates its own destruction. However, in the case of the other plasminogen activator, urokinase, it has no affinity for fibrin, but instead binds to cellular surfaces via a specific receptor called UPAR. And UPAR can act as an assembly point for UPA and plasminogen on various types of cells. And some interesting work that was published by a group by Di Vincenzio in 2010 showed that these fibrinolytic components need not be on the same surface in order to be able to activate one another and to potentiate fibrinolysis. So they performed a series of elegant experiments where they looked at localization of UPA and plasminogen on microparticles and on platelets and monocytes, and were able to show that there was crosstalk between various cellular components of the circulation. So from this work, at the similar time, we were looking at the ability of various plasminogen activators to lyse platelet-rich clots. And surprisingly, we saw a dramatic enhancement of platelets in promoting lysis in the presence of the enzyme SCUPA, single chain urokinase. And this was kind of surprising because our results with TPA and UPA were the opposite to this. So we took these observations a little bit further and we discovered that plasminogen was essential to be able to provide this stimulus in terms of enhancing SCUPA-mediated plasminogen activation. This was these graphs were done in collaboration with Lindsay Miles at Scripps Institute, and we compared SCUPA and looked at the rate of UPA formation over time in the presence of either wild-type platelets or from platelets from plasminogen-deficient animals. And you can see here that the platelets from the plasminogen-deficient animals were not able to stimulate UPA generation. We also looked at this as a function of various plasminogen concentrations in either a solution phase in the open circles or in the presence of platelets. And again, we see dramatic acceleration of UPA formation in the presence of platelets. So this then tells us that there's a reciprocal activation between SCUPA and plasminogen on the platelet membrane to be able to stimulate active UPA and active plasmin. This piqued our interest in looking at the various components of the fibrinolytic system in relation to the platelet surface. And at the time when we initiated these studies, there was many microfluidic models to look at the procoagulant capacity of platelets and subsequent fibrin formation, but none of these had been utilized to study fibrinolysis. So in collaboration with Johan Heemskerk's group, we adapted a model to be able to study fibrinolysis and to localize various components of the fibrinolytic system within thrombi. So we were able to see that, not surprisingly, given the fact that plasminogen binds avidly to the fibrin surface, we saw a large proportion of the plasminogen in direct co-localization with fibrinogen, or fibrin in this context, because it was in the presence of it had been clotted. But we did seem to see some component of the plasminogen that was directly in association with the platelet surface. To look at this more closely, we inhibited thrombin generation and subsequent fibrin formation with Herudin. And we're able to see direct localization of plasminogen on the cell surface, which we quantified with fluorescence intensity and saw an overlap in signal with DIOC6 from the platelets and with the fluorescently labeled plasminogen. So they then went on to look at direct binding of plasminogen to the cell surface, and this is exogenous plasminogen that we have fluorescently labeled and added to washed platelets. And we were able to see that when we stimulated platelets, we saw binding of plasminogen to the surface that increased over unstimulated platelets, and we saw the highest degree of binding of plasminogen was observed when we strongly stimulate platelets with thrombin and convulsin. This work that we have subsequently published in 2015 is not entirely new, because in the 1980s, Lindsay Miles and Ed Plough had looked at binding of plasminogen and observed binding to activated platelets. But we then saw that had to look more closely at the various subpopulations that were binding plasminogen, and we could see that it bound both spread platelets over the granulomer, and it also bound the procoagulant platelets, which expose phosphatidylserine on their surface, as denoted here with an XN5 in green. And as we showed in the flow cytometry results, if we increase the stimulus of the platelets, we increase the amount of binding, and that's very clearly shown at the bottom here, where we have a high degree of binding of plasminogen to that cap-like structure of these procoagulant platelets. We could attenuate the binding of exogenous plasminogen via incorporation of tirofiban, which inhibits that fibrinogen receptor, alpha-2 beta-3, and also by blocking fibrin generation with an analog GPRP, which will prevent any subsequent fibrin formation on the platelet surface. And that is you can see that both these inhibitors very avidly knock down the amount of plasminogen that is bound on the surface. We then turned our attention to thinking about platelet-derived plasminogen. We knew that there was potentially plasminogen that was sequestered within the platelets themselves, and we wanted to assess its contribution, to fibrinolysis. But when we set up an ELISA plate-based assay, when we looked at unstimulated and stimulated platelets, we did not see any difference in the amount of plasminogen. However, when we incorporated the lysine analog EACA, we were able to release plasminogen from the surface of these platelets, indicating that it was being retained on the membrane surface. We confirmed this with flow cytometry, where we looked at stimulated platelets, again stimulated with TRAP6 and convulsin, or with thrombin and convulsin in the absence or the presence of EACA. And in this context, we see when we look at the surface of the platelet itself, we see that plasminogen is directly associated with the platelet surface, and we can release that plasminogen when we include EACA, indicating that plasminogen is binding via a lysine-dependent manner. And likewise, similar to exogenous plasminogen, we see it binding in the cap structure of phosphatidylserine exposed platelets, whereas it's over the granulomer in PS negative platelets. Then begs the question as to how this plasminogen was associating on the surface of platelets. And plasminogen is fairly promiscuous in the number of receptors that it can bind, but the group of Lindsay Miles had identified a novel plasminogen receptor on the surface of monocytes. So we went on to study whether we could identify this same receptor on the presence of platelets. And indeed, we found that we could observe plasminogen RKT on the surface of activated platelets, and that there was some colocalization with plasminogen. There is not complete colocalization with plasminogen, because the antibody that's used to detect plasminogen RKT actually binds to the last 9 amino acids of the C-terminus, and thereby competes with plasminogen binding to the cell surface. To analyze this in further depth, my postdoc, Dr. Gail Morrow, went to the Scripps Institute, to the laboratory of Professor Lindsay Miles, and we studied the plasminogen binding in plasminogen RKT deficient mice and in plasminogen deficient mice, and we can see that we can see binding of plasminogen to the surface of the wild-type platelets, and this is attenuated on the surface of plasminogen RKT negative platelets. We can also co-localize this in terms of the confocal microscopy. What about, so the plasminogen's there on the platelet surface, can it be activated to functional plasmin? And indeed, yes, it can. So these are a set of fluorogenic assays that we performed on human platelets, and we looked at plasmin generation in the absence of any plasminogen activator. And when we stimulated platelets, we were able to see some generation of plasmin activity even in the absence of added TPA or UPA. And I should note at this point that heridin was included to inhibit any interference of thrombin in this assay. However, this was obviously increased when we included the plasminogen activators TPA and GPA. And you will notice here that there's quite a considerable difference between the TRAP6 and the thrombin in the terms of TPA, but not so much a pronounced difference in terms of UPA. And this alludes to the fact that TPA is very much dependent on that fibrin that's formed when we activate with thrombin, whereas UPA, as I mentioned earlier, is fibrin independent. We've also found that in some of our more recent flow-based models, we've been looking at the localization of TPA within the thrombus, and we find again that it coalesces with fibrin that is immediately adjacent to platelet aggregates, and in particular the fibrin that surrounds these platelets positive aggregates. And I just wanted to make mention of a paper by the group of Paul Kim, who has shown that activated TAFI, and this is the plasma pool of TAFI, will attenuate the binding of both plasminogen and TPA to the platelet surface. And he also showed in that paper that plasminogen and TPA compete for binding to the same receptors. So I've given you a lot of information there, and I just want to summarize this briefly, that PAI-1 and other inhibitors are abundant in platelets, and some of these are within the alpha granules. I should note that not all are in the alpha granules. TAFI is held within the cytoplasm, and the transglutaminase factor 13 is also within the cytoplasm, whereas PAI-1 and protease nexin 1 are found within the alpha granules and are released upon activation. We found that plasminogen binds both platelets bind both exogenous plasminogen, and they also retain platelet-derived plasminogen following activation. We found that binding is both indirectly via fibrin and directly via plasminogen RKT specific receptor. And we've also found that platelets have endogenous plasmin activity, and I haven't shown this today, but this graph alludes to the fact that the platelets themselves, which are shown in the purple line, can drive fibrinolysis in a purified assay. So why do sometimes platelets, why do they behave as antifibrinolytic, and why do they behave as profibrinolytic? And this is still a very much open question. We know that the composition of thrombi varies, and this can vary depending on the thrombus location in the vasculature. It can vary depending on the shear rate to which the thrombus is subjected. So you can see here that these platelets aggregates formed under low shear are very much rounded, whereas the high shear aggregates are almost like strings or little beads. And the fibrin that forms around them then replicates the structure of these aggregates. So this will be one various component. I've also shown you throughout the talk that not all platelets are the same. Some are PS negative, others are phosphatidylserine positive and have a very defined cap structure. So all these are different variables, and this is still an open body of work. So I think to conclude, I'll go back to my title, which is the interplay between fibrinolysis and platelets, and just conclude that it is very complex and still requires a considerable amount of work to tease out at what point a platelet can behave in an antifibrinolytic manner and other times switch on to being procoagulant. And this is not entirely surprising because a similar situation occurs in terms of the coagulation system. And it just remains for me to acknowledge various funders, particularly the British Heart Foundation, who have funded several project grants, and Claire Sally for many years, the Chief Scientist's Office, local charity Friends of Anchor, British Society of Haemostasis and Thrombosis, and also the university for the various facilities, the microscopy and the flow cytometry facility, and other members of the laboratory who have contributed to this work. Thank you for listening.